A very good day to Dr. Ajis. Today we're going to present our video presentation entitled General
Lab Safety.
My name is Elena Romulbanus.
My name is Nurnajdra Afifa.
My name is Nurfida Shafika.
And let's get started!
In any scientific context, lab safety plays an essential role because it forms the foundation for both individual safety and the accuracy of the experimental result. In addition to being required by law, maintaining a safe workplace is also morally right to safeguard our co-workers, ourselves, and the larger community. In this presentation, we'll guide you through the essential aspects of lab safety, focusing on the following key areas. Firstly, radiation safety. From the radiation safety, we can understanding the types of radiation commonly encountered in the lab, monitoring and minimizing exposure to radiation, managing risk with radioactive materials, using personal protective equipment PPE and shielding. Secondly, chemical safety. We can correct labeling and storage of chemicals, safe handling procedures to avoid spills and contamination, and safe disposal methods, emergency protocols for chemical spills and exposure. Thirdly, in the glassware handling, we can learn selection and inspection of glassware before use, techniques for safely assembling and disassembling glassware setups, procedure for cleaning and storing glassware to prevent accidents. Fourthly, general precautions. We can learn about general safety guidelines, electrical safety, heating safety, and guidance on handling using cleaning and care microscope.
What is radiation safety? Radiation safety is a set of safe practices and preventative measures implemented to ensure safety when working with or near radiation. Radiation hazards may arise from ingesting radioactive materials or radiation from external sources. The effects of radiation depends on the dose, duration, and the type of organ exposed. For the dose or quantity of radiation that was taken in, effects are usually more intense at higher dosages. For the duration, prolonged or continuous exposure over time increases the total dose and resulting damage. And lastly, for the type of organ exposed, different organs and tissues in the body have varying sensitivities to radiation. There are 4 types of radiation. First one is alpha radiation. Alpha radiation consists of 2 protons and 2 neutrons. It is also known as the helium nuclei. It is a positively charged particle. The effects of ingesting or breathing alpha emitters can seriously harm tissues nearby and increase the risk of cancer. The second type of radiation is beta radiation, which consists of electrons or positrons that are negatively or positively charged particles. The internal exposure towards beta radiation can increase the risk of cancer. Next, external exposure to beta radiation could also affect the skin and subcutaneous tissues. The third type of radiation is gamma and X-rays. Gamma and X-rays consist of electrons or positrons, which is negatively or positively charged particles. The effects of gamma and X-rays has the potential to ionize and harm genetic material and cells in the body. High doses of gamma and X-rays can lead to acute radiation sickness. The last type of radiation is neutron beams. Neutron beams consist of neutrons, which are uncharged particles, which can cause intense ionization along tracts, which damages the cells and DNA. ALARA stands for as low as reasonably achievable. What is ALARA? ALARA is a radio protection principle that limits ionizing radiation exposure to humans, animals, and materials while keeping diagnostic image quality. It foundations radiation protection. How do we keep the radiation exposure in terms of ALARA? The first one is distance. Keep as much distance as possible between yourself and the radiation source. By doubling the distance from the radiation source, reduces the dosage rate by 4 times in accordance to the inverse square law. The second factor is time. Work efficiently and limit your exposure to the source of ionizing radiation and leave the area as quickly as possible. And the last factor is protection and shielding. To reduce radiation exposure, always establish a barrier. Try to stand outside the room behind a concrete wall. And to prevent exposure and protect yourself from accidental exposure, wear gloves, aprons, and masks. Before working with radioactive materials, you should contact the radiation safety officers to obtain approval for use of a particular isotope. Next, you need to attend a radiation management training and obtain a radiation monitoring badge. Lastly, work using the standard operating procedure that has been approved by the Department Radiation Safety Officer. When working with radioactive materials, you should familiarize yourself with the nature of the radiation emitted, for example, alpha, beta, gamma, etc., by the isotopes and the tolerable levels of radiation for both short and long-term exposure. Next, never eat, drink, smoke, or apply cosmetics while working with radioactive materials. Never mouth pipette radioactive materials up into pipettes. Only use them as a safety device. Next, work with appropriate shielding and cover working areas with benchote, which is a polythene-backed absorbent paper. Also, use plastic trays that are clearly labeled to hold radioactive samples, pipettes, glasswares, etc. Moreover, you should mop up all spillages immediately using paper towels. Wearing a disposable rubber or polythene gloves while handling isotopes is a must. In addition, you should wear a film badge to help monitor radioactivity. And lastly, you should monitor radioactivity before and after every experiment using the handheld monitor where relevant.
In this slide, we can learn more about chemical safety. Firstly, general safety guidelines for handling chemicals. Secondly, specific precautions for handling acids and bases. Thirdly, safe techniques for smelling and mixing chemicals. And lastly, emergency preparedness. This is general safety guidelines for handling chemicals. Firstly, wear appropriate personal protective equipment (PPE). We need always wear lab coats, gloves, and safety goggles to protect your skin and eyes from splashes and spills. Use face shields and respirators when handling highly hazardous chemicals or when there is a risk of inhalation exposure. Secondly, avoid chemical exposure. Do not touch your face, eyes, or mouth while handling chemicals to prevent contamination. Then, wash hands thoroughly with soap and water immediately after handling chemicals. Keep chemicals away from food, drinks, and personal items to avoid accidental ingestion or contamination. Thirdly, proper disposal methods. Follow lab-specific disposal procedures for different types of chemicals. Know the correct disposal methods for solvents, acids, bases, and other hazardous materials. Use designated waste containers for hazardous chemicals and ensure they are properly labeled with the chemical name, concentration, and hazard class. This is specific precautions for handling acids and bases. When adding acids to water, always add acid to water first, never water to acid, to prevent exothermic reactions that can cause splattering and thermal burns. Then, pour acid slowly into water while stirring, continuously to dissipate the heat generated. When using fume hoods, we need to handle volatile or harmful chemicals, such as concentrated acids and bases, in a fume hood to avoid inhalation of harmful vapors. We need to ensure proper ventilation and regularly check that the fume hood is functioning correctly. This is the safe techniques for smelling and mixing chemicals. When we smelling chemicals, never smell chemicals directly to avoid exposure to potentially toxic fumes. Then, use the wafting techniques. Gently waft your hands over the opening of the container to direct a small amount of vapor towards your nose. This minimizes your exposure to the chemical fumes. When we mixing chemicals, add chemicals slowly and in the correct order to prevent violent reaction. Then, use proper mixing equipment and conduct the mixing process in a controlled environment, preferably under a fume hood. Read and understand the safety data sheets (SDS) for all chemicals before mixing to be aware of potential hazards and necessary precautions. And be aware of the potential for exothermic reaction and have appropriate cooling measures in place, such as ice baths or cooling jacket. Emergency preparedness. Lastly, spill response. We need to know the location and proper use of spill kits in your lab. In the event of a chemical spill, evacuate the area if necessary and follow the spill response procedures for the specific chemical involved. Then, use appropriate neutralizing agents for acid and base spills. Secondly, first aid. We need to familiarize yourself with the location and use of emergency eye wash stations and safety showers. In case of chemical exposure, flush the affected area with plenty of water for at least 15 minutes and seek for medical attention immediately. Thirdly, fire safety. We need to know the location and proper use of the fire extinguisher, fire blankets, and emergency exits. We need to be aware of the types of fire extinguisher available and which chemicals they are suitable for.
Why is glassware safety important? Because it reduces the risk of injury and accident. How can we ensure glassware safety? First, chipped or cracked glassware should not be used and shown to the lecturer. Broken glassware should not be disposed in the classroom trash can. Instead, it should be disposed in a special glass disposal container. Next, when pouring liquids into glassware, make sure the container you are pouring into is resting on a table at least a handbreadth away from the edge. Moreover, pour down a glass stirring rod to prevent liquids from splattering. If a piece of glassware gets broken, do not try to clean it up by yourself, but notify the teacher. When inserting glass tubing into the rubber stopper, apply a lubricant like glycerin to the glass and use a twisting motion. Lastly, do not place hot glassware in water because rapid cooling may make it shatter.
For the general safety rules, first and foremost, always listen to or read instructions carefully before attempting to do anything. Wearing appropriate attire is crucial. This includes safety goggles, lab coats, and closed-toe shoes. Make sure to tie back long hair and roll up loose sleeves to avoid any hazards. Keep your work area uncluttered and ensure you clean up the lab area at the end of the laboratory period. Be aware of the locations of emergency equipment such as fire extinguishers, fire blankets, IO stations, and first aid kits. And remember, report any accidents or spills to the instructor immediately. For electrical safety, always lay electrical cords when one can trip over them. Ensure your hands and the lab area are dry before using any electrical equipment. Never poke anything into electrical outlets and be sure to unplug all electrical equipment at the end of the lab period. When dealing with heating safety, allow burners and hot plates to cool down before touching them. Use tongs or protective gloves to handle hot objects. Always point the top ends of test tube that are being heated away from people, and never leave a burner or hot plate unattended. Next, microscope handling using cleaning and care. When handling a microscope, always pick it up using both hands, one holding the arm and the other supporting the base. Never grab or carry a microscope by its eyepiece. Place the microscope on a flat, stable surface. Before using the microscope, familiarize yourself with its various mechanical and optical components. Know what each knob does before operating it to avoid overworking or straining the mechanism. Use the microscope in a well-lit room or outdoors, and be sure to power on the built-in light before trying to use it for viewing. When using the microscope at its highest magnification, some models may require special preparation of the specimen or lens. For cleaning and care, always disconnect the plug from the main socket before cleaning the frame and stage. Clean these parts with a soft cloth moistened with a detergent solution, and ensure the instrument is dry before using it. When cleaning the optical parts, remember that the eyepiece and objective are coated. Always blow loose dust away first and use cotton swabs or lens tissue moistened with a lens cleaner or a small amount of alcohol. In conclusion, we discussed the importance of lab safety protocols, including radiation safety, chemical safety, glassware handling, personal protective equipment, the importance of knowing the locations and proper usage of emergency equipment, and the critical need to report any accidents or spills immediately. We also covered electrical safety guidelines, safe practices for heating and using hot plates or burner, and proper handling and use of microscope. Repeating the significance of these protocols, following guidelines is crucial to ensure a safe and productive working environment for everyone. Adhering to these practices not only protects you, but also safeguards your colleagues and the integrity of your experiments.
And that's all from us. Thank you.